Good afternoon. Oh, a lot of feedback. Hold on. Right, okay, it's Tuesday afternoon, week 4 of lockdown and of lockdown bioinformatics. So, welcome back. I hope everybody had a nice, peaceful Easter weekend. Managed to step away from any working from home that you're doing and have some time off. I did my best myself. And here we are again. So it's Tuesday, and we're going to change tack a little bit this week. So up until now, we've been getting ourselves familiar with the command line, learning some basic commands and how they work, how they operate, generally speaking. And that kind of culminated last week in us writing a not entirely straightforward script to download some data, build a multiple sequence alignment, and then on Thursday last week we looked at JALView, which was a multiple sequence alignment viewer, which we didn't go into a great deal of detail about, but which was a relatively useful piece of software for then looking at that alignment that we created. And so, in my mind at least, that brings us to kind of the end of the absolutely introductory material. So we've covered the basics that we need to be able to do some real work in anger. We've looked at the basic commands that we need by and large. There are some other commands that may come up as we go along, but I think we've learned enough now to be able to deal with those when they arise, rather than having to teach a whole bunch of stuff up front. We've also looked at scripting and how to begin to chain commands together kind of automatically, and that's essentially what we need to be able to do to begin to really do some bioinformatics in anger. And so the idea now, kind of week 4 that we're in now, is to begin to move beyond those basics and start to do some more complicated things. And we're going to begin, and this was my whole aim when I started this, was to take us through from those very beginnings to analysing some relevant RNA-Seq data. That was my kind of, the way that I pegged this at the beginning was that we would learn the skills that we needed to get us to a point where we could sort of organise that analysis ourselves, and that we'd have learned everything that we needed to learn to be able to undertake that analysis. We're now at a point where we can, I think, look to starting to do that, so that we can use the skills that we've learnt so far to download some relevant data. So download a data set that we want to analyse, and then to begin the processing of that data set using some command line tools. So I think those things that we've picked up over the last few weeks are going to enable us to begin that processing. What we can't currently do, what we haven't currently got as far as learning the skills for, is the slightly more complicated processing of analysis of that data once we've done the kind of preliminary processing, and that's going to be another amount of introduction that we're going to have to do once we've downloaded and processed the data. So what I'm going to propose that we do this week is look at getting the data downloaded and start to think about how we're going to process that data in batch at the command line, and then probably next week we will then begin to look at how we're going to analyse that data. And what that's going to mean for us practically next week is an introduction to the R programming language. So we're going to be using packages from Bioconductor for doing that analysis, specifically in the short term at least DSeq 2, which is used widely for analysing RNA-Seq data. But in order to make best use of those packages, like the same that we could have launched in 3 weeks ago to downloading RNA-Seq data at the command line, I didn't have to take 3 weeks to do all of the introductory material to get us to a point where we could download that data. But I think, and I hope that it's been instructive for everybody to do that, that we've got ourselves to a point where hopefully we understand a bit about what's going on when we're writing these commands. And I want to do the same for R. I'd like us to be in a position where we're not just kind of blindly issuing the commands that we're told to issue in R, but that we've learned a little bit about how the programming language works, and perhaps that we can begin to construct things for ourselves, rather than being completely reliant on example scripts and so forth. Speaking of example scripts, I've just realised that I have completely forgotten to do anything about putting my scripts online. I will do that. It's the perils of doing this as essentially my 1 hour of work a day at the moment, is that I'm on holiday. For those of you that maybe have missed me talking about that before, although that's probably unlikely if you're listening live. But I'm currently taking a break from work because my plan pre-lockdown was going to be to take the 2 weeks of the school Easter holidays off, and I saw no pressing reason to change that plan once the lockdown happened, because various domestic reasons that I'm not going to trouble you with. But so I'm currently on holiday. I'm not doing any work other than this live streaming. So therefore, I haven't had the time to devote to getting things like those scripts online, but I will do that, I promise. Certainly before the end of this week is out, because I think that will be useful for people to have a place to go to be able to see my scripts outside of the environs of these little videos. So okay, so that was a sidetrack. What was I saying? Yes. So spending time with R, yes. So I'm going to introduce R in much the same way as I introduced the command lines. We're going to work from the basics, and we're going to take it relatively slowly to get us to a point where we actually analyse the RNA-Seq data. Expect that's kind of what to expect over the next couple of weeks. So this week we're going to download the data, look at processing the data. Next week, then we're going to look at introducing R, and I think that's probably where we're at. So with all of that in mind, let's have a little bit of a think about RNA sequencing before we think about anything else. So as I stated right at the outset, my plan was to analyse some RNA sequencing data. And the reason for that, and there's lots of reasons for that, it's a relatively self-contained task. In terms of training, it's the thing that we've done the most in the training that we provide in the Bioinformatics Support Unit. So we have run training courses. I think I talked about this right at the outset, but we have run training courses in my unit for, I think, more than 5 years, and particularly for the last 3 or 4 years, what we've really focused on is the kind of material that I've been presenting so far, these kind of the introductory things, and the kind of example that we chose for that was RNA sequencing, because it's fairly self-contained. You can teach a discrete set of stuff, and you can do useful things to get some RNA-Seq data analysed. It's also the data which we have in the unit, we have the most practical hands-on experience of. We've analysed lots and lots of different types of data over the years, hundreds and hundreds of experiments, all different types, every sort of sequencing experiment that you can think of, pretty much. But the thing that we keep coming back to again and again, and the data that people keep bringing to us, is expression data, and that has been the case ever since the unit started in 2005. We began by doing an awful lot of microarray analysis, and certainly I had done a lot of microarray analysis over the years, and obviously in the last 5, maybe 7 or 8 years actually now, it's really started to shift over to RNA-Seq, and I don't think I've touched a new microarray experiment for 4 or 5 years now, and I think it's been all RNA-Seq. During that time, that shift really has happened and kind of been completed, really, at Newcastle at least. And so we have, as a unit, and me individually, but as a unit as well, we have an awful lot of experience analysing RNA sequencing data, and so therefore it seems like a very sensible thing for us to try and teach. It's the thing which we probably understand the most deeply. So that's kind of where we are. When we started 3-4 weeks ago, I was looking around at data sets and there wasn't a lot out there, so I was thinking, well, maybe there's a SARS data set that we can use, maybe there's like a flu data set or something like that that's maybe tangentially related to COVID-19, but actually since then, because science is moving apace and people are publishing preprints and data sets and all sorts, there is actually a SARS-CoV-2 data set, human tissue cells in tissue culture infected with SARS-CoV-2 virus. There's a data set on GEO, on the gene expression omnibus, that we can hopefully download and take a look at. My one caveat to that is I have not yet tried to download it, because as with all of these things that we've been doing, there is a certain amount of live testing going on. So we will explore the data set together, and we'll explore how easy it is to get hold of that data set together. Certainly I've downloaded one of the files, so I know it's possible to download some of the data, and I know the means by which we're going to do that. So that's what we'll kind of move towards doing today. But first of all, I wanted to do a little bit of an introduction to RNA sequencing for those of you who are perhaps not overly familiar with it as a technique. Maybe a technique that you've heard of and that you're aware of, but that you haven't really dealt with in anger yet. And so I wanted to spend half an hour or so just in introducing RNA sequencing as a technology and what some of the implications are for it, for the analysis that we'll be undertaking. So with that in mind, I will almost certainly draw some stuff as we go. So I'm going to get my whiteboard. And I wanted to show you a picture of the day because I feel like it's a feature that's lapsed slightly. But that said, again, Jacob's been busy in the way that 7-year-olds are busy today, so I don't have a Jacob picture of the day. But my older son has been spending some of his holiday time doing YouTube drawing tutorials, and he's very keen that there's a Noah's picture of the day. So today we've got Noah's picture of the day, which is a dinosaur that he drew last week following a YouTube drawing tutorial. So for context here, Jacob is my youngest is 7, Noah is 12, so obviously his drawing skills are perhaps a little bit more advanced, but he's very proud of this picture, and that's, I guess, is quite understandable. It's quite a nice picture, really. So that's our picture of the day today, not a Jacob picture of the day, but a Noah picture of the day. Okay, so I'm just going to get my whiteboard working because it seems reluctant to sync up today. Then when is it ever not reluctant to sync up? Let's face it. So just waiting for it to refresh, and I will be with you. There we go. Maybe that's going to work. Right. Yay, we're on. Right, okay. So I will obscure Noah's picture with my whiteboard, and off we go. Okay, so RNA sequencing. So let's just make sure that I'm oriented on my page here. There we go. Right, okay. So RNA sequencing, yes. Why did that do that? Sorry, I'll stop talking to myself and start talking to you guys. Right, okay, so so RNA sequencing is the use of sequencing to profile, discover, and quantify RNA. So you extract your RNA from your whatever your system is that you're looking to sequence, that you're looking to understand. Sorry, and you convert it to cDNA. Then you directly sequence that cDNA. So in doing that sequencing experiment, you not only are you profiling the RNA that's present. So if there's RNA in your cellular system, it should result in reads in your sequencing library, and so you can use that sequencing library to profile the RNA that's present in your system. But also, by virtue of the way that the sequencing library is constructed, you also quantify that RNA. So it's a quantification technique, a relative quantification technique, as well as a profiling technique. It has, as a technique, has by and large replaced microarrays. I think, as I talked about our experience in Newcastle, which I think is probably not completely atypical for us. RNA sequencing has completely replaced microarrays for expression profiling. There are still some applications for microarrays. It's not a completely obsolete technology, but for expression profiling, we're entirely RNA sequencing based now. And as a technique, it has a lot of advantages over microarrays, particularly in terms of dynamic range, and also being able to use it for non-standard organisms as well, for organisms where we don't necessarily completely understand the genome yet. We can, even in those novel organisms, we can still use RNA sequencing to profile gene expression. So it can be extremely useful. It's the methods for analysing RNA-seq data underwent extremely rapid evolution from kind of the advent of RNA-seq. So the first RNA-seq paper was 2008, so just over 10 years ago now, where it inherited almost entirely analysis methods from microarrays, to now where we have all sorts of specialist techniques for analysing RNA sequencing data in and of itself, but also analysing different aspects of RNA sequencing data. So not just looking at gene expression, but transcript expression and allele-specific expression and all sorts of other things besides. That evolution of analysis has perhaps slowed down a little bit now as the technique has become more mature, but I think we can still agree that RNA sequencing is an extremely versatile technique, and the analysis does have many subtleties, and that's part of what I want to draw out over the next couple of weeks is the subtleties in the analysis, and not just the straightforward A to B to C methods for analysing that data, but what the subtleties are, and how we kind of try to address those a little bit. So I'm going to talk a little bit about the technologies that we're going to use for our analysis, just in kind of overview, and where we're going to start is where all of this always starts, which is with some data. Okay, so here's my data, and for RNA sequencing, the data is sequencing data, so it's in the standard sequencing data format, which is this FASTQ format, which I'll talk about in more detail when we have some examples of it in front of us, and when we talk about FastQC, perhaps later this week. So FastQ is the standard data format for sequencing data. For the experiment that we're going to be analysing, we need to download that data. So we've not done a sequencing experiment, but we're going to download that data from sorry, my brain's a bit of brain fade. We're going to download that data from an online repository. So we're going to download from the Gene Expression Omnibus, or GEO. And the actual sequencing data itself is stored in the sequence read archive, the NCBI sequence read archive. So in order to download that data, we're going to be using the SRA Toolkit, or SRA Tools as it's referred to. So so this is our software for downloading our data. So whereas in an ideal world, maybe we would want to use the tools that we've already learned about, so wget, for example, which we've used for downloading data already. In an ideal world, we would continue to use those tools, but the SRA have to distribute so much data that they found that those tools don't really scale very well for the amount of data that they distribute. So instead, we're using SRA tools to do it. So I'm not the world's most expert user of SRA tools, but we'll introduce that for downloading our data. So then we want to, once we've downloaded that data, we want to do a couple of things with it. So we want to do quality control, which I will talk about in much more detail in a subsequent lesson. But QC is extremely important. You should never undertake any high throughput analysis without a reasonable understanding of the QC processes that you need to understand how good that experiment is, and how the quality of the data that you're putting in to your analysis techniques. So we'll look at some QC later on in the week. The other thing that we want to do is we want to quantify that data. So we want to undertake a process of quantification, which will produce count tables. For this, there's lots of options. Mostly these days you would use software like Callisto or Salmon, which do quantification at the transcript level. So these are tools which are very similar to one another, both in terms of the results that they produce and the speed with which they produce them. These will give you your kind of derived data set from your input FastQ, which is would be your count data. And for our purposes, for the experiment that we're doing, this will be whichever software we choose to use will produce transcript level counts. So instead of counting gene expression, we're going to count transcript expression, and then we analyze at the gene level. So we want to transform that count data from transcript level counts to gene level counts. So it's still count table, but this time it's gene level. and we do that with an R package. So this is where we're going to step into R. We do that with an R package called TX import. So that's where we step into R, and then the final thing that we're going to do is then analyze that data. So we're going to analyze using R and bioconductor, and specifically DE-Seq 2. So that's our kind of our software here is these specific things, and here for QC we're going to use fast QC and multi QC in the first instance at least. So that's the software that we're going to use outlined in red there. So we've got quite a few things to get to grips with, quite a few things to learn. Why do we do things this way? Well, it's a well-established pipeline of tools. It's relatively modern but robust. So the software that we're using here has all been around for a few years and it's well developed. And in most multi-method comparisons, this looks to be about the most accurate way of doing this analysis. And what I don't want to do, so when we first started doing RNA-Seq courses, we used to teach everything at the Linux command line, and because we didn't teach any R at all at that point, for various reasons, we used to do the analysis using the Tuxedo suite, which you may have heard of. So it's an aligner called Top Hat, and then a quantification and analysis tool called Cufflinks. And that worked very well as a command line tool, but Top Hat was developed right back at the beginning of RNA-Seq analysis, really. And as the world moved on, it became not the most accurate way of doing the analysis, and so we had to move on as well. And so what we teach now, and what I'll be teaching over the next couple of weeks, is, in our opinion, kind of the modern, robust way of doing this analysis. That's not to say that things won't change in the future, but this seems like a reasonable pathway for analysis right now. What I do when I teach RNA-Seq, both for students, so for undergraduates and postgraduate students, but also on courses for researchers, is I give a bit of a glossary of terms. Some of this stuff might be kind of self-evident to you and obvious, but some things are there's quite a lot of terminology that gets thrown around in sequencing experiments generally, but in RNA-Seq as well. And so I just like to kind of clarify a few things. So we'll do a bit of drawing, and I'll try and clarify as many of these terms as possible as we go. So obviously, when you're doing RNA sequencing, you're doing functional genomics, right? So you're working with the genome in some way, shape, or form. And we're working with the products of the genome, but nevertheless, that's our kind of raw starting point, is the genome. So that's the genome, and obviously, when we're thinking about RNA sequencing, we're thinking about the genes that we find in the genome. So here's a gene in our genome, which so we're going to be working with human human experiments. So those genes are organized into introns and exons. So we have our gene has exons, exon, and introns, and our DNA has, we can think about the DNA in the region of a gene as having a coding strand which is the gene on which we find the gene on, and the template strand, which is the opposite strand. This is context specific, so if our gene was on the other strand, then the coding strand would be the bottom strand and the template strand would be the top strand, because it's a context specific thing. This here is our gene. Okay, so we get RNA produced from our gene by a process of transcription. So transcription produces mRNA. Okay, so that's the product of our transcription. In this particular case, we've got two. Bear in mind, we've got two exons, so we've got a splice junction there. So we've got exon 1 and exon 2. And then, for the purposes of our sequencing, we take that mRNA and we break it up into fragments. So we randomly fragment our mRNA, and those fragments, because we'll have multiple molecules of RNA, so those fragments will be overlapping, because you'll fragment your different molecules of RNA differently, so you end up with fragments of mRNA, which you then turn into cDNA. So the next process that we need to talk about is reverse transcription, which takes our mRNA fragments and converts them back into cDNA, so cDNA synthesis. So this would be double-stranded molecule of cDNA eventually. And then, so this is, so here we've got, just checking this is going to appear on my screen, this is fragments. So they're quite often referred to as fragments, and even once you've done the sequencing and we're thinking back to the thing that we've sequenced, we tend to be sequencing fragments. Once you've converted to cDNA, and importantly, you've made your sequencing library, so you've stuck your adapters on the end of your cDNAs, then what we have now, which was a fragment, we now refer to as an insert, because it's inserted into our into our sequencing library, and then these things here are adapters, and they contain barcodes and primers, primer sequences, and all sorts of other things that allow us to actually do the machinery of the sequencing. So lots and lots of these constructs together. So if we just get rid of all of this for a second, so if you've got lots and lots of these inserts, lots of drawing. Apologise for my scruffy drawing, it's nowhere near as good as my children's. So again, these all have adapters as well. You get the picture, right? We get lots and lots of these inserts with their attached adapters. And this is what we refer to as a sequencing library. Okay, so because we've got lots and lots of different inserts all ready for sequencing, this is our sequencing library. Okay, so that's usually referred to as a library. And I can see that my screen isn't necessarily keeping up with all of my deleting, so I'll catch that up in a second. Okay, so that's our sequencing library. So let's just get rid of what I've drawn so far. Get rid of all of this. And then we'll talk about what we do with that sequencing library. So that library goes onto a flow cell, onto a sequencing flow cell. The case of the experiment that we're going to be analysing, I think it's the sequencer that's been used is a NextSeq, so an Illumina NextSeq. But there are lots of options. So that's what you do with your library. So you stick it on your sequencer, and from that library, so remember we've got we've got our construction. Here we go. Okay, so that's our individual piece of cDNA that we're going to sequence. So this is our insert that we're going to sequence. From that insert, we generate sequencing reads. Okay, so we generate two, usually or often, a pair of reads, although not always. And actually, in the case of our experiment, I think we're just generating single-end reads. But you generate your reads from the two ends of your insert. So from one end going in one direction and from the other end going in the other direction. This is dependent on your sequencing primers. These reads are often, they used to be referred to as forward and reverse, or F and R. So the first one that was produced would be referred to as the forward read, and the second one would be referred to as the reverse read. But actually, there's not necessarily an orientation implied. So forward and reverse don't necessarily make sense. So actually what we generally talk about these days is first in pair and second in pair. And that hasn't come out very well at all, has it? Okay, so first, is that better? Not really. Love this. It's excellent when this technology works, but not great when it doesn't. Okay, you get the idea. First in pair, second in pair. And that's how we generally refer to the reads. So what you will often see in the names of your sequencing files is you'll get the sample name, so sample, and then somewhere in the file name you'll get the lane of the sequencer that it was sequenced on, and then you'll get either R1 or R2, and R1 indicates that that's the first in pair read, and R2 will indicate that it's the second in pair read from that particular lane. So that's what we're thinking about when we think about certainly the file names that we get back from our Illumina sequences in particular. And do bear in mind that this is almost everything that I'm referring to here is referencing Illumina sequences, because that's the majority of the data that you're going to see when it comes to RNA sequencing in particular. An awful lot of this data will come from Illumina sequences. So that's your sequencing library, and so you generate your reads from your sequencing library. In general, in an RNA sequencing experiment, you will generate millions of reads from your sequencing library. So you'll start with probably billions and billions of fragments. There's a certain amount of selection that's involved in generating your sequencing library, and then from those fragments you will generate millions, certainly millions, sometimes often tens of millions of reads from your sequencing library. So that's the general process. The one thing I should say, I've talked about with the first in pair and second in pair reads, I mentioned that there isn't really any implicit orientation, but actually with RNA sequencing there often is. So I'm just going to talk about strandedness at this point. What you will often find, so if we go back to our genome organization with our gene here, I'm just going to refresh. Bear with me a second. I'll put Noah's picture back. Just try and catch everything back up again to each other. Okay. 1 second while this refreshes, and I'll just quickly show you what I mean, what I'm talking about in terms of orientation. So for RNA sequencing, there actually is an orientation in a lot of experiments, because the way that the library is constructed means that you have strandedness to your library. And what that means in terms of the experiment, what you will find is that your first in pair read, so if we go back to our genome here and remember that this is this is coding, the top strand here is coding, the bottom strand here is template, and we were going 5' to 3' like this, so the gene is effectively going in that direction. What you'll find with a stranded RNA-Seq experiment is that your first in pair read will map to your genome, it will map to the template strand going in the opposite direction to the gene, and your second in pair read will map to the coding strand and it will be going in the same direction as the gene. So actually, in a stranded RNA-Seq experiment, and most RNA-Seq experiments in 2020 are stranded because most generated using the Illumina TrueSeq protocol, which is a stranded protocol. So what you will find is actually there is an implicit orientation to the reads in a stranded RNA-Seq experiment, and your first in pair read will map to your template strand going in the opposite direction to your gene, and this really helps you in circumstances where you have overlapping genes. So I'll talk a little bit more about some of the other concepts that we're working with RNA-Seq as we move through doing the analysis. So that's kind of my introduction to the basic idea for RNA sequencing, and then what I now want to do is talk a little bit about the data that we'll be working with. So we're going to download this data, like I said before, from the Gene Expression Omnibus, and we're going to be using SRA tools to do that. So there's a couple of things that I want to do before we finish today. So the first thing is that we need to set up a new Conda environment for our new experiment, and I would like to install SRA tools into that Conda environment. And then the second thing that we need to do is I just want to show you an example of downloading one of these files. And then tomorrow we will look in more detail at the data that we're going to be downloading, and we will get together a list of the files that we want to download, and we will get to actually downloading that data. It's going to be there's going to be a reasonably large amount of data. So the nature of sequencing experiments in general is that they produce a large amount of data. So we need to go into that bearing that in mind. And we are going to be downloading gigabytes of data, not megabytes. So now is the time to consider whether you have the space available for that or not. It's likely to be, I think, somewhere between 10 and 20 gigabytes of data. So we do need to think about that a little bit as well this week, which we haven't really run into before. So for the first time this week, I'm going to go to my terminal. So I'm going to open my VM, and I'm just gonna close everything down from last week. And actually, I'm just going to do that by rebooting. So you can reboot from the command line. I'm not sure if we've done this yet. Because you installed your virtual machine, you are a root user, you're an administrator on your virtual machine. So you have admin privileges, and you access those admin privileges at the command line by prefacing your command with this executable called Pseudo. So Pseudo is the thing that says what Pseudo does. It takes the following command, so whatever comes after Pseudo, execute that command as an admin user. Okay, so I'm going to run Pseudo, to say, okay, I want to be an admin for this one command, and then I'm going to run shutdown. So shutdown is a command which does exactly what it says on the tin. It shuts your computer down, and it takes options which modify its behavior, like so many other of the commands that we've been working with. An important one for us in this instance is -r, which tells shutdown not just to power the machine off and leave it off, but to power it off and reboot it. So -r means reboot the machine. And you need to tell the system when. So you can schedule this. You could say in 10 minutes or in an hour, but we want to do it now. So we want to shut down, and we want shutdown to reboot the machine, and we want to do that right now. There is a command called reboot, which somebody has mentioned in the comments. So you could do sudo reboot now. The reason that I don't do that is for one whole release of Ubuntu, reboot was broken and didn't work. And when you try to reboot, your machine wouldn't come back, which is fine for a virtual machine, which you can just prod and say and press go on again. But for a remote server, which you don't necessarily have physical access to 24/7, reboot not working is an absolute disaster. So I got into the habit of using shutdown to reboot machines. So that's why I do that. But there is a command called reboot, which may well work. So sudo shutdown -r now, and that will prompt me for a password, because it wants me to confirm that I really am an admin user. And so it wants me to know that it wants me to enter my password. When I type my password, nothing appears. So there's not even any asterisks or anything like that. There's no prompt that you're actually typing anything, but that's expected. And then hit enter. It is possible to mount an external hard drive on a VM. I'm looking at the live comments again. It is possible to do it. We installed the guest editions for VirtualBox, which I think is one of the things that you need, but you also need the host editions to be able to mount a hard drive from your host machine onto your VM. It is indeed possible to do, and I have done it in the past. There are almost certainly very good instructions online for doing it, and we can probably track those down if there's lots of people wanting to do it. Right, so I've rebooted my machine. I felt like it needed that. I've got the software updater is sitting in the background, which is going to prompt me to install updates. I will probably do that between today and tomorrow, because it makes sense to keep your machine up to date if at all possible. Some of those updates will be security updates. Okay, so terminal emulator is what I want, and that drops me into my base Conda environment. I want to create a new Conda environment for doing our RNA-Seq things. So I'm going to do Conda create -n. So we did this last week, and this is the way that we create a new environment in Conda. So Conda create -n, and my Conda environment is I'm going to call RNA-Seq. So this is where we're going to be doing a lot of our software installations over the next couple of weeks. Okay, so creating myself a new Conda environment. This will just take a few minutes, and will ask me to confirm that I definitely want to do it. So yes, I'm happy for that to be where you put this environment. And then it gives me the instructions for how to activate it. So Conda activate RNA-Seq. So if I highlight that with my mouse and then middle click, it pastes it in at the command line, and that will activate my RNA-Seq environment. So the first piece of software that I want to install in this environment is SRA tools. We don't need to worry about channels at this point, because we've added the channels to our Conda config, so that's true across all of our environments. So we've already got our channels set up, so BioConda is available to us, so we don't need to worry about channels. We just need to do a Conda install. So Conda install SRA tools. Okay, so that's our command to install the SRA Toolkit for ourselves. So the SRA Toolkit, for one thing, has a fair number of dependencies to make sure that it works, which is not massively surprising. It's there you go. That's done really quite quickly, and I should now have all of the tools that are in the SRA Toolkit available to me. So the SRA Toolkit actually consists of lots and lots of executables. The one which we really care about for downloading data from the SRA is called fastq-dump, fastq-dump, and we should now find that that's in our path. So there you go, -H to give us the help. So that's the help information for fastq-dump. And now we just want to test that it works. Okay, so we're just going to build a single fastq-dump command for one SRA entry, and it is actually going to be one of the entries that we're interested in, but that doesn't really matter right now. I'm going to do this on my shared drive because otherwise it's going to occupy too much space. So I'm going to cd share, and then I'm going to make a new directory, and it's going to be week 4 because that's the week that we're in, and then cd into week 4. And then we're going to run fastq-dump. So we've got a new empty directory. We're just going to run fastq-dump to download one single file from the SRA, and then we're going to take that what we've learned for that one file, and we're going to build up, as we did last week, write a script to download this time from the SRA rather than from Uniprot. So let's have a look at the SRA, the fastq-dump help just quickly. So fastq-dump has quite a lot of options, and that's because data could end up in the SRA, in the sequence read archive, from any number of sequences. Basically, any sequencer that's been manufactured, there could be data from that sequencer in the SRA. So it has to be able to deal with all of those things. The way that data is stored in the SRA is not actually in fastq format. It's stored in what are called SRA files. And so what fastq-dump is actually doing is converting from the SRA file back to fastq. So it's quite configurable. It's got a lot of options. And you can see all of those options here. And you've got options for splitting out, and that's for paired end reads, where you don't want just one file, but you want your first in pair reads in one file and your second in pair reads in a second file. It's got options for filtering and for clipping reads, for getting alignments as well as for raw reads, and for producing compressed output and various other things as well. In our case, the use case is relatively straightforward, which is the reads that we're going to download right now are single end reads, so we don't need to worry about splitting them out into separate files or anything like that. They are they're Illumina sequencing reads, which means that pretty much the defaults work for us for Illumina sequencing reads. And we don't even need to worry about, as I said, dealing with multiple files or dealing with multiple samples or anything like that. So the usage is relatively straightforward. So fastq-dump, and then options, and then the accessions that you want to get from SRA. So the one option which I'm going to give two options. So fastq-dump, so that's our executable, that's our command, and then the options that we're going to give it, although it's not a recommended option, I'm going to do it because I don't like writing uncompressed data to the disk. So I'm going to produce my output is gzip compressed fastq, so I'm going to give it the --gzip option. And the other thing is that by default, we'll talk about the fastq format in more detail another time. But by default, FastQDump will write the sequence identifiers into each of our sequences twice, and we don't want it to do that because it's wasteful of disk space. So we're going to format this defline qual. So that's the option that we want, defline qual, and we want it to just be a plus symbol. That's all we want in our qual defline. So that's me determining the format of that definition line for the qualities to get rid of it printing that identifier twice, because that's wasteful. And then finally, all we need then, so this will output to the present working directory, and it will output based on the name of the accession that you give it. So we're going to give it an accession number, SRA 114122. Why is that not done now? 11412215. Okay, so that's the sequence read archive accession for one FastQ file for a single run. So we will have to download lots of these to get all of our files that we need for our whole experiment, but we're just going to download one today just to show that it works. Okay, so I'm going to execute this command, and it's FastQDump is very quiet, so I'm going to hit enter, and it's just going to sit there and look like it's not doing very much. So that's because it's downloading a very large file. But unlike wget, which gives you a little progress bar and tells you what it's up to, FastQDump doesn't, not by default anyway. So this is just going to sit here for a few minutes while it downloads this file. So I'm just going to grab a second terminal just for a second. So I'm going to open a tab. So that new tab opens in the same working directory as I was in my other tab. So if I type ls here, we should see that what was an empty directory now has a FastQ file in it. It's a growing FastQ file. So if we do ls-lh, we can see that currently is 28 megabytes. And if we run it again, it's probably grown a bit. So yes, now it's 33 megabytes. So it's going to keep growing while it downloads. If we want to have a peek at what's in there, we can't use cat or less because it's compressed. So we asked for gzip compression, which means that it's downloading it as a compressed file. So we can't use cat or less because what we will see is gobbledygook. And I'm just going to prove that. It's always I'm going to take my life in my hands, and I'm going to prove that it's gobbledygook by using less. Oh, less has been clever. How has less been so clever? Okay, let's be unclever. Yeah, there you go. So cat will show you the binary. So that's because it's compressed. So it's no longer a text file. It's now a binary file. So cat is showing us the binary, which is gobbledygook to our eyes. Less was very clever, and I wasn't aware that less could be that clever, but less showed me the contents of the file. I'm used to using zless for gzipped files, so zless will let you see through the compression to see what's in the file. It seems that less, and maybe it's bash being clever and working out that this is a gzip file, so it sent it to zless, but zless will show you the contents. And so this is our FastQ file that is downloading. So it's not finished yet, and it will take a little while. I'm just going to leave it downloading after I've signed off, but that file now is downloading, and you can see it's getting bigger. So now it's 99 meg and still growing, still downloading. So that's the initial process of downloading that FastQ data, and I will talk a little bit in a little bit more detail about FastQ when we've downloaded more data and we look at FastQC. So tomorrow we will look at the experiment and find all of the SRA accessions that we need to download all of the data, and we'll write a script to download that data and leave it running overnight, and hopefully it'll take less than 24 hours, hopefully, fingers crossed, to get all of the data that we need so that we can then proceed with doing stuff with that data on Thursday. Okay, so but with that I will sign off for today. I know that it's been a lot of talking today and less doing, but we will have more doing tomorrow. So it's important to get these introductions done. And I don't just want to proceed blithely as though everybody gets what an RNA-seq experiment is and how it works. Okay, so I will come back tomorrow and we'll look in more detail at getting more data from the SRA. But until then, have a safe evening, and I will speak to you all tomorrow. So, sayonara. Bye, everybody.